Hi guys, good morning everyone. And today we have as a host, I already told you, Jairo Mendoza Roldan, which is a friend and a colleague. He comes from Colombia but works in Bari University. And besides being passionate of reptiles, he also works on these animals, in particularly in with synanthropic and also wild animals. And he works mainly in looking at zoonosis, so parasites can be transmitted directly or with vectors from animals to humans through reptiles. So, Jairo, welcome and feel free. Everybody, feel free to raise questions, and he is very friendly. And for everyone that could be interested in making some thesis with reptiles, he is the right person, and will be also here in presence in
For
June.
sure.
And so have a good lesson. I will be following you, but I have some commitments also here for some research field, and so have a have a good lesson.
Thank you, Antonio. Good morning, everybody. Firstly, I would like to again thank Antonio, a great friend and colleague, for inviting me to talk about one of these subjects I am really passionate about, as he mentioned, which are reptiles and specifically their parasites. So today I would like to tell you more or less the last 4 or 5 years of research we have been performing in the University of Bari, where I am currently located and working. My experience with reptiles comes from when I was in Colombia, and I'm a veterinarian degree, and I performed a master's and a PhD on epidemiology of ectoparasites of reptiles in Brazil, and afterwards here also with zoonotic diseases in Italy. So what I would like to show you, actually, given that this is a very wide, as reptiles are very diverse, their parasites are very diverse as well. I would like to show you some of the new research possibilities, new perspectives, and specifically what are the ways we can study to understand the relationships between reptiles, humans, and their environment, as well as their parasites, and how then we can associate and study their parasites. There's specific ones, of course, that are part of their microbiota, but as well as those that are and can have a zoonotic potential, considering also now the paradigmatic example of the possibility of pandemics and spillover from pathogens coming from wildlife and to human populations. So I will try to tell you, or I will show you some of the new studies we've been performing in many aspects, always following that idea also of wildlife mainly, because reptiles as well as other type of animals, we have different types. No, we can study them from an ecological perspective, from animals that are kept in collections or zoos, as well as animals are kept as pets. So I will try to show you more of what the role these animals may have as synanthropic animals adapted to urban settings or wildlife, which is, I think, the area you're currently studying, of course, under the umbrella of One Health. So I will try to keep always that concept within the different studies. In any case, as Antonio mentioned before, if you want to discuss or ask anything in any moment, you can always stop me or raise your hand and we can discuss. So this can be a really easygoing conversation between us this morning, so we can discuss also about, as well as Antonio also mentioned, I will be going there again to Sardinia, which I loved because I was there a couple of months ago. And we can discuss if you are interested in any of these type of studies for your thesis or for future projects. In any case, if we would like to talk about reptiles and their parasites, we initially we will need to discuss about where the area of they are involved in, which is what part of veterinary medicine. That part of veterinary medicine that specifically discusses or treats and studies reptiles and their parasites is herpetological medicine. Pathological medicine includes not only reptiles, but also amphibians. So it's a very wide area of the veterinary medicine, which has different specializations. Currently, of course, I'm working with parasites, but there are other areas in which a veterinarian or physician can work on as other animals. So we will have internal medicine, pathology, surgery, neonatology. So there are many areas in which a veterinarian can specialize, but as well as other areas of veterinary medicine, it requires experience, but also requires the understanding of the reptile or the species we are working on because of their great diversity. So the first home take message of this conversation of today is that reptiles are a group of animals which are incredibly diverse. So we will have different type of animals and different morphologies and physiologies that will thus need a level of specialization, also requiring, if we were going to work with these animals under the veterinary medicine concept, or even with their parasites. Under this, of course, as we need to try to specialize again on a specific part of these animals, I choose to study the herpetological parasitology, which is an area, of course, indeed, also very wide and very diverse, considering the different type of parasites we can find even in the domestic animals we are used to treat or to study in the field of veterinary medicine. But when we talk about reptiles, we are talking about a big, wide diversity of parasites. In any case, before talking about their parasites, let's discuss a bit about the reptile patient or the reptile within veterinary medicine. When we discuss this, we need to consider the different types of reptiles that can arrive to our clinical setting or for being assessed by a physician. So then we can have a different type of classification. So we can discuss or group this type of animals by different types of reptiles. And of course, every type of reptile will have a very different setting of specialization, also of clinical examination and of treatment, considering also the relationship this animal can have with its tutor, or not having any tutor at all, just having a relationship with the environment and thus with the human settings near that reptile. So we can classify these animals as pets, which we will have an intrinsic relationship with a person. That will be the person that will give us the anamnestic data of this animal, and afterwards also the husbandry issues and other type of history of this animal, including age or previous treatments, type of morphology. So we will have a bunch of important information that will give us the opportunity to assess properly these animals. Differently from if we have animals that are coming from zoos or collections, because we still have a lot of information, but of course, these animals will be less adapted to being handled because they will be in a different setting. The collections maybe can be given by a breeding program for reintroduction of some type of a species of reptile, or they can be from a zoo, so they have a more educational and other type of goals for these animals or objectives. So they will be treated in a different way, and of course, these animals will have a less interaction with humans, even in the case that most likely they will have an ID number, not a name, for example, but we will have a lot of information regarding this animal and how we can then use the medicine-based evidence or evidence-based medicine to treat and assess this animal. And the group in which we will have less information, but we will have a wider understanding of the relationship between reptiles parasites are those that are considered wild, synanthropic, and importantly, as the example that you are seeing in this picture, which is a Burmese python, invasive species. I put this example because one of the species that is now considered invasive and very widely distributed in Florida, in the United States, is the Burmese python, which was introduced because of the reptile market of pets, then introduced accidentally, and now it's very largely distributed around the Everglades, causing, of course, an ecological distress and the reduction of some species, native species, and importantly, the spillover of some parasites. For example, these animals are really commonly infested or infected with pentastomids that I will show you afterwards. And there has been some spillover to native species of snakes. And of course, in synanthropic animals that are more adapted to urban settings, well, we will have an interaction in which there could be some type of spillover or transmission or even encounter with this animal. So we will have different types of reptiles. But before considering what are the pathogens and parasites we can transmit, let's look at these animals as our patients. How can we classify and how can we actually assess a reptile? We first need to know some basic information or characteristics these animals have. So initially, we need to consider that reptile patient in general will be an ectothermic animal, which means that they do not control their core or body temperature. They depend on the environmental temperature, and they generally are regarded as having a preferred optimal temperature zone in which these animals can thrive. There are some exceptions, but in general, though, all of the reptiles are ectotherm, and this implies that our animals that are adapted to temperate, to tropical and subtropical temperatures. So the colder, the less species we will have. And also that they will have a lower metabolism that we see in mammals or other endothermic species of animals. Thus, the metabolism is very involved in many of their physiological behaviors, reproduction, and even the way then we will treat or assess or evaluate these animals, for example, for surgery. On the other hand, reptiles have different strategies of reproduction. So they generally are oviparous. Generally, most of them, of reptiles, still lay eggs, which allows them not only to have a great number of clutches, I mean, in terms of 10 to 20 or more eggs per clutch, but to be a strategy that is even less it requires less energy and metabolism for the species to be able to reproduce. Whereas for some species, such as, like, for example, vipers and other snakes, they are viviparous, which means that they have a higher mammotrophy or placentation, and which implies a longer period of gestation, but also a higher energetic intake and metabolic intake. So this implies that these animals can reproduce only, for example, one time per year, whereas reptiles that use still oviparous strategies, they can reproduce 2-3 times depending on if they are in tropical or temperate regions, such as, for example, Europe. But something that almost all, if not all, reptiles share regarding their reproductive strategy or their life cycle is that all reptiles are precocial or nidifugous. That means that once they are born, they are completely independent. Most of them, with some few exceptions, for example, crocodiles that have a maternal instinct and take care of their younglings for at least a couple of weeks. Most of reptiles do not have parental care. So reptiles, once they are born, they are completely independent, and they rely on phenotypical instinct for already knowing what they need to do and how they need to hunt and all of their behaviors. And also, importantly, their morphology is the same once they're born and once they become adults. So they can change, for example, in color, or they can change in patterns of the skin, but their morphology, I mean, for example, a baby snake, once it's born, for example, a boa snake, it's born in a certain morphology. It will be the same morphology, just smaller than an adult. And this is the case for practically all reptiles. They do not suffer any type of metamorphosis between the time they're born in which they are adults. They depend on metabolic intake, nutrition to grow, and of course environmental factors such as temperature, humidity. So a reptile that is in good conditions, I mean, at least through a environmental standpoint, they will grow and be in their form as a smaller animal, but once they are again adults. So that what is implied that once an animal is born, they already have to face all of their survival instincts and of course predation. So I will give you an example of a very dramatic, I will say, video I like to show, because it's the predation instincts of two species of reptiles in the Galapagos Islands, in which once the marine iguanas, which are endemic from the Galapagos Islands, once they are born, they're already faced with the need to escape from snake predators. And they are completely independent and alone in this sense. So enjoy. So this is to just illustrate how reptiles are precocial and completely independent, again, once they are already after born. Other characteristic reptiles may share, or at least we can classify them, and of course, depends on this characteristic, we will have different ways of assessing the reptile health. Is, for example, reptiles have scales, but they are not always the same. Some are covered, so the squamata, for example, which means scales, but also we can have plates or osteoderms, which are, for example, in the case of turtles, the turtles are actually the carapace. They are not inside a carapace, for example, as snails. They are, the carapace is part of the turtle bone structure and also skin structure. Whereas, for example, crocodiles, as you see in the right picture, they are covered with osteoderms, which means that they have a scale that is composed by in the same composition as bone and also skin and connective tissue. And most of reptiles, they have a shedding system, which means that allows them to molt or shed their skin eventually and with certain frequency. But the real, I mean, the group that actually sheds their skin is squamata. It's a suborder. We have Reptilia as a class of reptiles, and a suborder which is squamata within the order Reptilia. That they actually within the up order Squamata, which actually has two suborders, which are Ophidia and Sauria, which they actually shed their skin, and this shedding of the skin can be complete in the sense of snakes, where they completely have to shed all of the skin in one occasion, or incomplete, as in the case of lizards and other type of sauria, in which they lose part of their skin and then the skin comes out by parts. They have a very particular anatomy, most of reptiles, with, of course, we have different characteristics depending on the group. One of the things, for example, that it varies from the anatomy that we're used to seeing, for example, in mammals and birds, is that they have a three-chambered heart, which means that they have a more primitive yet successful circulatory system, in which we find one ventricle and two articles. In this sense, the circulation is less efficient in one way, but it has other possibilities. For example, they can produce a left side shunt of the heart, which allows them to go through voluntary or physiological apnea. So many reptiles that are used to being underwater for a long time, more than 2 hours, they can completely shift their circulatory system to go around their body, with the exception of the lungs, so they can recirculate already oxygenated blood and allow them to, of course, remain without breathing for a longer time. This is important, for example, if we're considering animals that have to go under anesthesia. So, of course, for example, marine turtles, aquatic animals will be less prone to go under induction if we use only volatile anesthesia. Of course, another thing that is different from other animals or other classes of animals is that they have not a division of their body between thorax and abdomen. They only have one cavity that is called coelomatic cavity, which, for the exception of crocodiles, that they can seem the more primitive, but they are more morphologically and anatomically similar to mammals than other reptiles. They have 4 chambers and they have a pseudo diaphragm. So with the exception of crocodiles, most reptiles have only a coelomatic cavity, in which the respiration movements are given by ligaments and muscles that are connected to the wall of the coelomatic cavity. The differences, for example, if we take for the example of snakes, we can see that most of reptiles have the glottis in the anterior part of the oral cavity, and most reptiles will have a non-parenchymatous lung, which means that they will have a sacculated lung that ends in an air sac, and they have a distribution as a beehive, which is composed by the functional structure, which is called faveoli in reptiles, and the gas exchange between CO2 and O2 is given in this irrigated part of the anterior part of the lung. If in the air sac, it is just to accumulate for more time the air. And another a characteristic from reptiles is that they don't have a mucociliary system, which inhibits or doesn't allow reptiles to cough. So they will accumulate exudates and liquids within the lung And that's why most of them, when they're suffering from some type of respiratory syndrome or disease, they will have upper or respiratory tract infections. They will present themselves with mainly open mouth respiration and stargazing position. Other thing that is in hindrance of the assessment of the health of reptiles is that they don't have visible sites or veins for venipuncture. So we have to allow or understand the topographic position on some of the veins. For example, in this case, you're seeing a ventrococcygeal vein blood draw, which is with the right after the body of the vertebrae, of the coccygeal vertebrae. They do not have, with the exception of snapping turtles, they don't have lymph nodes, so they have a very different lymphatic system. In this sense, they have always, in most of the bigger vessels, they will have both blood vessel and a lymph vessel, which will have an issue when we are discussing the problem of hemodilution. So every time we have to draw some blood, we need to be very aware that possibly we can have hemodilution, and then our results and hematic evaluation of a reptile has to be taken with caution to avoid that. For example, we can have animals that we can consider them with anemic, but actually it's just lymph hemodilution. Also, something very important is that when we see the blood, that differently from mammals, they still keep their erythrocytes nucleated. They don't lose the nucleus. This allows them to live for longer. So an erythrocyte of a reptile can live over a year. But of course, they will have a less capacity of regeneration. So many times when we have anemic reptiles, their erythropoiesis is not so fast as the one we see, for example, in mammals. And something that's very important, apart from the very different morphology of their cells, of their blood cells, depending on the type of reptile, is their hemoparasites. So already we see that we have a very large diversity of hemoparasites. This is a study from Brazil, from West Brazil, in which you can see different parasites from the intra-erythrocytic and plasma parasites, such as, for example, trypanosoma or microfilaria, and some artifacts. So we already have the first insight on, or second take-home message is that diversity of parasites in reptiles, as is already from this group of animals, it's quite high, and most of their parasites may be already commensal parasites, which means that they do not produce any deleterious effects on their typical host. And this is given by the convergence of evolution or coevolution of parasites with their typical host, which occurred millions of years ago when these animals diverged or they originated. They already had a relationship with some type of parasite that, by coevoluting to different specific species, now, for example, as hemogregarines, as you can see, hepatosomgamons, they now are almost commensal. Some of them are even can have a positive relationship with reptiles. Some nematodes and protozoans from the gut of, for example, herbivorous reptiles are needed for the digestion of cellulose and other nutritious intake of elements. So this is very important, because when we identify a parasite in a reptile, and more importantly in a wild reptile, the important thing to take into consideration is that most likely we will not need to treat this animal, because it's part of their microbiota or of their parasite fauna. So we need to consider this, because if we take parasite identification, parasite treatment. In reptiles, it doesn't work that way. We need to assess if, in fact, this can be a deleterious relationship or not. And most of the cases, reptiles will have 80% of their parasites will be innocuous or will be already a commensal part of their intestinal or body fauna. With that said, considering parasites, then if we are considering that most of them can be commensal parasites or non-pathogenic parasites, I have been studying or tried within the concept of herpetological medicine to assess or to put together or to have a conversation between different factors or different concepts. The main one, of course, parasitology, meaning assessing the parasites this specific group of animals may have, but then integrating or having a more holistic approach, considering other two elements or two important factors. The relationship with humans and their environment, which we can study under the umbrella of the One Health concept, and the zoonotic parasites and zoonotic diseases and pathogens that these animals may have a role within the epidemiological life cycle of these pathogens, and how they can relate to human and environmental health, for example, as sentinels or bioindicators of environmental health, as well as the risk of infection given our relationship with these animals. So the concept of One Health is very important. It's an approach which has increasingly have a more pivotal role within our studies and our role as veterinarians. As veterinarians, we within this concept have a stakeholder role and a decision-making role, given that we are the union between human health, environmental health, and animal health. One Health, regardless, is a multi-disciplinary approach. So we will have biologists, medical physicians, politicians, people working on chemistry, people working on many areas, and us as veterinarians are let's say a point very in the middle where those concepts may join. And this will give us then insights on how we can study these relationships between humans and reptiles. Why is this very important, or at least why I think our studies have been undertaken within the concept of One Health? It's because we're facing, or we are in a historical age, which is called the Anthropocene, in which the herpetofauna is, of course, suffering from a lot of anthropic pressure. The anthropic pressure is given because of our increasing human population and our behavior and relationship within our environment and our planet Earth. And this is given by mainly three types of anthropic pressure, which are deforestation, the increasing urbanization, and the fragmentation of the habitats our ecosystem these animals live in. For example, you can see in this map how the projection and how it is now the urbanization in Italy, in the Italic Peninsula. Most of the territory now already is urbanized, so it's divided in different small villages, cities, and anthropic areas where, for example, they are used for agro and animal production. So already we have a lot of areas that are already being used for some type of anthropic and human development. If we have an increase in this and fragmentation of the habitats, of course, animals need to go somewhere, no? So they will go to an area where they can thrive, where they will find shelter, food, and they will find peers of their own species, so they can, of course, reproduce. So we will have an increase in synanthropy. Synanthropy is exactly this: when a species of animal is very plastic and adaptable and can go to a peri-urban or urban area and find what it needs to thrive, we will have an increase in numbers, and we will have an increase in places where these animals may be. Of course, we have animals, for example, in this case, rattlesnakes in South and Northern America, or other species of reptiles living in sympatry and in synanthropy in urban, or even, for example, here in Italy, you can see commonly Moorish geckos or lizards. Then we will have an increase in the possibility of encounters and, of course, accidents. In one hand, domestic animals, carnivores such as dogs and cats, can increase their level of predation if they have outdoor behaviors, which means that they can play a role in the trophic change of transmission of parasites and pathogens, but even also have accidents with venomous species or poisonous species, for example, some toads or amphibians. And regarding people, we can have more encounters that will allow us, or unfortunately, to have more accidents. And of course, this will imply venomous or non-venomous type of reptiles. When we have all of these factors together, then we will have, of course, the increasing possibility of having also transmission of diseases or pathogens and parasites. And something very important is that already a neglected tropical disease that is considered to have more than 100 people dying of this disease, tropical disease, that still doesn't have is neglected because it doesn't have the proper attention by governmental governments and politicians, is snake bites. Snake bites in tropical and subtropical areas is still one of the most important diseases, with, of course, more than 4,000 people having some type of disfigurement or disability after the snake bites. So it's still something we need to work on, even in, for example, in the Mediterranean area. But of course, there's always a positive aspect of studying not only the venomous species, but also the venom in per se of these animals, which is the possibility of having compounds of this venom that can be used for different biotechnological and pharmacological applications. Indeed, there are some studies that show that venom of some snakes, for example, vipers and cobras, lapid snakes, they can have some peptides that can have an effect or can act against protozoans, such as Trypanosoma cruzi for Chagas disease, Plasmodium for malaria, and even Leishmania. So there's always a possibility of protecting and having conservation initiatives of these animals to be able then to study the venom and have pharmacological applications of the venom of these snakes, apart from trying to find different strategies to control and to eradicate and to avoid the consequences of snake bites. So now coming to fruition in terms of the conversation of today, when we are discussing about taking into account all of these factors about herpetological parasitology, as I mentioned before, once I have a reptile that I need to assess for any given situation, and most likely can be given by a parasitological disease, I will find a myriad of parasites. Most of them, as I mentioned before, will not have any type of negative effect on their host, but some of them can be pathogenic to the reptile, but even they can have a zoonotic concern. Thus, that is the area in which our studies from the last 4 to 5 years have been taken into account. And this is also considering that the parasites of reptiles, they can be divided in some way in two categories, depending on the relationship with their hosts. One, they can use reptiles as intermediate or paratenic hosts, such as, for example, some helminths, in the case of, for example, cestodes, for example, Spirometra, that uses reptiles as hosts. Then they are eaten by, for example, humans, and they can be transmitted even to people. And those that use reptiles as definitive hosts, as, for example, some mites and ticks, and pentastomids, the one that you are seeing coming out of the glottis of this Federlands snake. Also, we have another group of parasites that can be also of zoonotic concern, considering also vector-borne pathogens, which is ectoparasites, and they can be divided in if they use the reptile host as a permanent host or as a temporary host. So we have as permanent parasites Acarina, the class Acarina, subclass Acarina, in which we can find mites and also ticks. In this case, you're seeing a picture of a nymph of amblyoma, with also larvae of trombiculid mites or chigger mites in a snake. And also another very important group are temporary parasites, such as mainly dipterans, composed by sandflies or phlebotomin species of sandflies, and mosquitoes, which reptiles can be even parasitized by leishmania or be host of arboviruses transmitted by mosquitoes. So considering all of this diversity of parasites, those that can be transmitted to humans will be also depending on the type of interaction we have with reptiles, because, of course, the more intrinsic this interaction is, the greater the possibility is of transmission. So we have a group of reptiles, as we saw before. Pets, reptiles are generally, thankfully, they come from captive breeding, so they are bred in conditions that reduce the level of pathogenic parasites and pathogens, but they still may have some parasites that can be transmitted transtadially, or, for example, if they have an outdoor enclosure, they can be infected with some type of parasite. So there's a higher or more intrinsic relationship between humans and pet reptiles, but there are less parasites associated with this if they come from captive breeding. If they come from sometimes reptiles that are kept as pets, they are captured in nature and then brought to captive conditions. This happens most of the cases, for example, with snakes from Asia, and they come with their parasites. They enter in a stressful event, which is being in captivity and being handled, and then the immunity reduces, and they can be then infested and have pathogenic effects of these parasites, and afterwards they can transmit, of course, these parasites to humans. Also, in some cases, parasites, the interaction we have with reptiles is in very different herpetological situations. For example, reptiles can be eaten or used as part of the culinary culture of some regions. This can be associated, I think most of you will think, this is or Asia or Africa, but this picture over here actually is from my hometown near Barranquilla in Colombia, where it has a religious also connotation, because on the Holy Week in Colombia, people are not allowed to eat red meat. So many people eat fish, and also animals that live in water, or animals that have scales. So many people eat, especially in impoverished areas, they eat reptiles. So this can be associated to the transmission by the trophic way or pathway of some parasites. And the area which I am more focused on in the past years is the relationship with synanthropic animals, which of course is a involuntary relationship we have with these reptiles that can have a positive effect because, of course, for example, if we consider Moorish geckos that they are around urban and in urban areas, they feed on many insects that can and arachnids that can be harmful. So they have a important role also as synanthropic animals, but they also can have some parasites that can be transmitted by indirect contact with humans. We already seen this, sorry. So we can classify these relationships or these pathways. We did this review once I came back to Italy in 2019, in which we summarized the main pathways reptiles may transmit parasites that can be through oral ingestion or ingestion of raw or uncooked meat of reptiles. For example, for trichinella, for some parasites such as Sarcocystis or other type of nematodes. And then we have also an environmental contamination, which is related to reptiles that live in urban or peri-urban areas, or people going to wild settings. And this is the case most likely of some type of protozoans and the pentastomids. And the one that we are studying the more for the past years is the one transmitted by vectors and parasites. Of this case, we can see some cases of leishmania associated to reptiles that increasingly we're seeing that there are some species that could be also transmitted to humans. When we consider in zoonotic diseases, the most likable disease that or pathogen that we can think of is Salmonella. And indeed, reptiles are asymptomatic hosts that excrete intermittently this bacteria. There are many serovars that are associated to reptiles, and they are associated mainly to people that have them as pets. But it's not only Salmonella, the parasite that we can see associated to reptiles. Indeed, other parasites, for example, are helminths. What you're seeing here in the lizard, apart from the pentastomid we already saw, is Mesocestoides, which is another type of cestode that can be ingested by lizards and then transmitted by the predation to dogs, cats, and in some cases, unfortunately, also to humans. Indeed, one of the areas we also are studied was the possibility of transmission of zoonotic parasites through depredation. Of course, us as veterinarians need to be very aware of the impact dogs and cats, if we are working with these animals, may have on the endemic fauna of certain areas where people used to have these animals living outdoors, and they can predate many type of small animals and even in some cases large animals, and be infected with the parasites, and these animals have a role as intermediate or paratenic hosts. Some of them are not zoonotic, but some of them can be transmitted then if we are in contact with the feces of these dogs and cats, or even if we, for example, cats are used to or have a behavior of bringing some gifts to their tutors, and then if we are in contact with that prey, that unfortunate animal that was predated, sometimes they can also be transmitted by the contact with this small prey. For example, the one I mentioned before, if we consider reptiles as intermediate hosts, one of the of pathogens that can happen, of parasites can be transmitted, is Mesocestoides, which in general is not that pathogenic because dogs and cats are definitive hosts, and cestos, as you may know, adult stages, they generally don't have a very high impact or big impact on their definitive host. apart from, for example, nutritional imbalances or anemia. But in some cases, the larvae, the tetratheridium of Mesocestoides can go through the intestinal wall to the peritoneal area and then produce ascites, and they can continue to replicate within this area. So what you're seeing here is peritoneal liquid full of tetratheridia. And this, of course, is something that can produce a lot of deleterious effects and pathogenic effects to the host, but it can even also be transmitted, in this case, if not taken into consideration. And it's very difficult to treat once these animals are the only option can be praziquantel, and sometimes it doesn't even work. So it's an emerging issue, the one of Mesocestoides. Also, for example, definitive host reptiles can be part of the epidemiological change of pentastomids. What you're seeing here are adult pentastomids of Rayetela. Rayetela is a pentastomid of lizards and geckos. This was the first time we observed it in geckos from the Mediterranean basin. And generally, they live in the lung of these animals, snakes and lizards, and then they release the eggs within the oral cavity, and then they ingest the eggs, which are excreted, and we can observe then in the feces the larvae, the embryonated eggs, within the feces that are infective to even humans. And what is the way of easiest way of transmission of this pathogen? Also, predation. So we describe this pathogen from Linosa. It's a pathogen that was mainly associated to North Africa and Asia, and it can produce in humans a disease called creeping disease, because once you ingest the larvae or the eggs, if they are embryonated, they will migrate through the skin, producing a type of sensation. That's why it was given this name. And it's because people sometimes can eat unawarely feces, or even sometimes people eat raw geckos in some areas of Asia. And the main reason of transmission or to urban settings is predation of given mainly by cats. So that's why the third, I think, take-home message of this discussion or conversation is we need to, as veterinarians, be aware of the impact dogs and cats have in the environment, and always advise and recommend tutors of these animals to keep them indoors, giving them the proper stimuli indoors, but not allowing them to go to outdoors and produce depredation, deleterious effects. So the reptiles, when we assess them, we need to also take into consideration where the parasites may be. For example, we need to assess the oral cavity. That will give us a lot of information regarding the parasites they may have, and sometimes we can find unexpected parasites, such as the case of mites or ticks, in this case Ixodes ricinus, in the mouth of a four-line snake here in Italy. And this is because as reptiles shed their skin, sometimes they shed their skin and they will shed, of course, their ectoparasites. So there's a strategy on some ticks and mites of infesting the oral cavity, in which they can, of course, remain for some time, given that it's a semi-anaerobic condition, so they can thrive and survive at least for a couple of days. More in depth regarding the reptile vector-borne diseases, we also, after having that main or first review on zoonotic parasites of reptiles, then we decided to go more in depth of what is the main area of study of our group here in Bari, which are vector-borne pathogens and parasites. So I wanted to study first which will be the impact, or what are the parasites and pathogens that may be transmitted. To study this, we need to first consider what are the main parasites. So they can be mites or ticks, and sandflies and mosquitoes. Once we have this taken into consideration, we can see that the ones that have a direct impact, not only because because they are permanent parasites, are mites and ticks. And mites and ticks of reptiles, they can be classified mainly in three large groups, which are Acariformes, under where we can find the order Trombidiformes, that I will show some of them. These are the less pathogenic species of mites, because, as I explained before, they have had convergence evolution or coevolution with their hosts. So some of them already have the morphology of, for example, scales. In the case of this is a Jacobia hemidactyly, imitating a scale of a gecko. And then we have the real parasites or parasitiformes, in which we have exotic ticks, argacid and exodide ticks, and we have mesostigmata mites. Of course, one of the main issues, if we consider mites and ticks, is the infestation of reptiles, is the hyperinfestation. Of course, if we see the snake, this is a Morelia spilota in Australia, parasitized by Botrocodron idiosauri, a type of tick that also infests mammals. And this, of course, is not a happy snake. It's a snake that is suffering from edema, most likely dehydration and anemia. And this can bring the animal to die if not properly treated. And this is a wild animal, so this is a hyperinfestation, producing an important deleterious effect. But we also can find other direct effects, such as dermatitis in some species, an immune response given by cellular-mediated immune response, and liberation, for example, of histamine, anemia, as you can see here, and this ecdysis, which is the improper molting of the skin or shedding of the skin, or as you can observe here in this rainbow boa, and respiratory signs. This is given because some of the parasites of mites are lung mites of the Entonicidae family, so they can infest the lungs of these animals and produce many clinical signs, such as stomatitis, stargazing position, in which the snake is almost drowning because it's full of liquids, its lung, and pneumonia. In some species where we have some structures, such as the laurel pit, it can be impacted given by the accumulation of mites and ticks in that areas, impairing them afterwards to, for example, predate properly their food. And especially and importantly, there are many pathogens correlated or related to the infestation and then thus transmitted by mites and ticks. They can be divided in viruses, bacteria, and protozoa. I will give an example mainly of bacteria, but just to also mention an emerging virus of snakes, mainly transmitted by mites, is Ophionitis natricis. It can also be transmitted by direct contact or vertical transmission, but it seems that Ophionitis natricis, which is the snake mite, has a very important direct role in the transmission as a vector, and it produces these neurological signs. It's a reptarenavirus, which it produces a disease called inclusion body disease, which has no cure, and it has a very negative prognosis. And within the bacteria, we can find many vector-borne bacteria that also may have a zoonotic implication, such as, for example, Rickettsia and Borrelia, which is the Lyme disease. In one of the first studies we did was to try to understand the parasitic fauna. When I was in Brazil, so we studied and we published this paper in which we studied different areas of Brazil, different states, where we tried to understand and identify the main species of mites and ticks. and then their pathogens they might transmit. What we found is that most of the areas where we collected, we found mainly here, this is the greener area, the darker green is Sao Paulo state where I was located. So of course, we had a bias in sampling animals, number of animals, because there was the area which I was mainly collecting, but we also had other areas coming from the northern part, such as Pará or Acre, in the north part of Brazil, here near the Amazon. And we found many mites and ticks, for example, larvae of trombiculid mites that also infest humans and other animals, oribatid mites that are phoretic. We also found many type of ticks and mites of different, many different genera. And we were able also to see where they were located. So mainly they were located in and infested reptiles in areas where reptiles first could not eat or take out the mites or ticks. And secondly, in areas that in some species are called mite pockets, because they're specific areas where they concentrate. And of course, as I mentioned before, it seems to be an adaptation of the host and the mite. In one case, to avoid dermatitis and other negative effects on the host, and in order to protect in some way mites, as you can see, as they are all associated to one part of these lizards. We also find many type of ticks, for example, soft ticks and species that can be also infesting humans, such as Amblyomma sculptum, and the typical reptile ticks such as Amblyomma dissimile and Amblyomma rotundatum. Then those mites and ticks we studied molecularly to identify some molecular, some vector-borne agents. I mean agents because some of them, of course, are not pathogenic. So we found two main groups, hepatosome or hemogregarines, which are specific for reptiles, but we also find rickettsia, which is the spotted fever group disease. Here you can see where we found mainly in the southeastern part of Brazil and in the northern part of Brazil, associated to snakes mainly. Hepatosome, we found many type of parasites that are already associated to different species, and they can be transmitted to predation, mosquitoes, or even also mites and ticks. But importantly, rickettsia, we find some species that could be pathogenic and transmitted to humans. Considering this, that they were also captured or collected in urban and peri-urban areas, further highlights the importance of understanding which are the implications and role reptiles may have in the perpetuation of some zoonotic bacteria. Then when I came to Italy, one of the first studies we performed was to try to understand if in southern Italy, we had also here in Basilicata and Puglia region, we had also the occurrence of Borrelia burgdorferi sensulato, which is Lyme disease. That it's one of the main bacterial vector-borne pathogens in the northern hemisphere, and we did find it in Ixodes ricinus and in the Neotrombicula autumnalis. You can see here a lizard, wall lizard, co-infested with both. We found Borrelia in Ixodes ricinus, but not in Neotrombicula autumnalis. So that then we thought another type of pathogens, such as rickettsia, what we had studied before. No, so we went to study also considering that we have a spotted fever group associated to the Mediterranean area, and in that sense, we try to study that pathogen. But before that, then we did another review specifically for vector-borne pathogens of associated to reptiles, and what you can see here is that we have negative effects. We have possibly positive effects of some parasites transmitted by reptiles, and we have others that are just mechanically transmitted, such as, for example, Ariomonas from mites. From ticks, we have rickettsia and Borrelia. So we already mentioned Borrelia, and I will then mention a bit about Leishmania, which is the one that can have a possible, and then I will try to explain why positive effect on mammals. So this is the areas where we generally studied vector-borne bacteria of reptiles. So areas that were peri-urban, not so urban, but where we had ticks and mites associated to reptiles. And one of the areas we studied was the Parco Regionale di Gallipoli Cognato and delle Piccole Dolomiti Lucane, which is this park here in Basilicata region, a mountainous area full of natural settings, reptiles, and of course, ticks, as for example, this Dermacentor and this Yaloma. So we studied these ticks and mites from wild reptiles and synanthropic reptiles associated to natural settings where people generally went to practice outdoor activities or people working on those areas. Of course, then considering the One Health paradigm. So we studied the reptiles associated to those areas where people usually frequent them and work, for example, forestry workers or farmers. And what we found is that reptiles, those that are in those areas, their mites and their ticks, for example, Isolis ricinus, are infected with rickettsia. And also humans are exposed to those pathogens. So this closes the concept of in areas where these animals may perpetuate or be infected with these pathogens, they can serve as reservoirs and maintain some species of Mediterranean spotted fever. To end up or to wrap up the part of vector-borne pathogens, I would like to show you the recent studies and perspectives and highlight some of the future studies we are thinking on is considering leishmania and reptiles. I think most of you and many people do not associate leishmania with reptiles because we generally associate leishmania with mammals, mainly dogs, and with sandflies that feed on mammals. But of course, there are some sandflies and also some of them occurring in Italy and the Mediterranean basin that feed on reptiles, such as the one you're seeing here in the middle of this picture. In this case, many species of leishmania are associated to mammals, but some of them can be associated to reptiles, and they are distributed mainly in the Old World, for example, in the Palearctic area, in the Mediterranean basin, in Africa, and some of them in Asia. If we see the classification of leishmania, we can see that we have between the real leishmania, I mean Euleishmania, we have 4 subclades. And within these 4 subclades, 2 of them are strictly or co-evoluted, or they diverge at the same time. So they are considered sister clades, which are leishmania, that includes 18 of most of them pathogenic species that use mainly phlebotomus and Lutzomia sandflies to transmit this leishmania to mammals, mainly canids, rodents, and in some cases humans, as some species are anthropomorphic. And there is another group of non-pathogenic, I want to highlight this part, non-pathogenic leishmania called Sauroleishmania, that includes more or less 18 species, distributed mainly in the Old World, I mean Mediterranean basin, Africa, Asia, that are called Sauroleishmania because they mainly infect lizards and geckos, and they are transmitted mainly by another genus of sandflies called Sargentomaya. In this case, in Italy, we have Sargentomaya minuta. The important thing is that actually Sauroleishmania could be involved also in human leishmaniasis or zoonotic leishmaniasis, such as for the case of Leishmania adleri, which is associated to a lizard called Latasia longicaudata in Kenya and in another parts of Africa, which was considered to be non-pathogenic until it was seen that it can infect macrophages from rodents and humans, and some cases of cutaneous leishmaniasis were associated to this species of leishmania. If we go here and locate ourselves in the Mediterranean basin, we have from both groups of subclades of leishmania, we have some representative species, mainly Leishmania infantum, which is associated or distributed around the Mediterranean basin. And we have another species of Sauroleishmania, mainly Leishmania tarentule, which is also occurring practically in the same areas where Leishmania infantum is. If we then focus further on in Italy, we can see that Italy is an endemic area for Leishmania infantum. In the recent studies, it's been seen that observed that all of the peninsula is endemic to Leishmania, not only the south that was considered before to be endemic, but the south is hyper endemic, which means that more than 60% of dogs are exposed to this parasite. And despite human cases being underreported, there are some cases of imported Leishmania to the Italian peninsula, but most of them still are considered exotic. So in Italy, we have Leishmania infantum, but we also have another species that is called Leishmania tarentule, that, as its name mentions, it's from the typical host is Tarentula mauritanica, or the Moorish gecko, which is this species of gecko I think most of you have seen running around urban areas. And it's transmitted by this sandfly, which is Centomaya minuta, which feeds mainly, but not only, on reptiles. It's a species that we have observed that it actually has a peripilarian type of development. What you're seeing here is the Malpighian tubbles of sandflies infested, which means that they are in the lower or in the hindgut of the sandfly, but they also migrate to the anterior part, to the stomoidal valve, and they also replicate and invade this area, which means that they can be also transmitted through the blood meal, after the blood meal of a sandfly. So we have both of these species occurring in Italy, but meanwhile, for Leishmania infantum, we already have a lot of information regarding its biological life cycle, its their typical host, its other non-permissive or non-permissive hosts, and of course its zoonotic implication and transmission. For Leishmania tarentule, we still have many doubts and many gaps in its life cycle. But there is something important that you can see over here, that it's that, okay, it can infest, and generally its cycle involves reptiles and sandflies, but it can be also possible that this Leishmania can infect humans and other mammals. This is in a nutshell, given because of I think you have a question. What symptoms does Leishmania manifest in this species? It's non-pathogenic, so generally it doesn't have any clinical sign, not even to its typical reptilian host. And this is something that I want to highlight, and I will explain later because why it can be then having a positive effect even in the mammal host. So Leishmania tarentule in Italy, it was described firstly in Taranto, in Monopoli, which is this town, this town over here, a seashore town in Apulia. And it occurs in sympatry, this is important, with Leishmania infantum. So endemic areas for Leishmania infantum, we also have the occurrence of Leishmania tarentule. And it was described in the Moorish gecko, and in this also the Cosci gecko here, also from southern Italy. And it was seen the amastigote-like forms infesting other type of cells. So we are used to seeing amastigote forms in mammals in the macrophages. In this case, they were found or in the erythrocytes and even in the thrombocytes of these animals. We performed then different studies to see where it was occurring, Leishmania tarentule, but we also had the objective to see if reptiles, if they could be infected with Leishmania infantum, given that we have to consider an endemic area for a pathogen such as Leishmania as not dog, sandfly, humans, Leishmania. We have to consider all of the factors that can be involved within that epidemiological context. So we also consider reptiles living in sympatry in, for example, dog shelters, urban areas, or parks in which we have the other elements of the epidemiological cycle of Leishmania. So we wanted to see in the observing the four southern regions of Italy, namely Apulia, Basilicata, Calabria, and Sicily, in synanthropic reptiles, not those that are more forested, but those that are more occurring in peri-urban or urban areas, if they had molecularly positive samples for Tarentule, but also for infantum. So we had the authorization. This is an example of lizard lassoing from an island in Sicily from, I think it's Filicudi, where then after sampling some of the animals, we took blood samples, and from others we took all the tissues, so they were ethically euthanized and humanely euthanized, and then we took the samples of different tissues to see where they had Leishmania, and we also performed cytology. And what we observe is that for almost 300 squamata reptiles, I mean snakes and lizards, almost all of them were occurring in almost all of them were moriscos, these ones you see here, and they were from dog shelters and peri-urban areas. And we did found Leishmania tarentule, of course, in 26 of these animals, 10%, but we also did found some of them with Leishmania infantum, further implicating reptiles as possibly other natural reservoirs in endemic areas for Leishmania infantum. Of course, this is a preliminary result. We need to further address and isolate actually Leishmania infantum from reptiles in order to say that they are natural reservoirs of and amplifiers of Leishmania infantum. But on the other hand, we also started to observe some interesting circumstantial evidence that humans may be infected by Leishmania tarentule. The first was a study in Brazil, where they find some molecular traits of Leishmania tarentule in mummies, 300-year-old mummies. But then in Italy, firstly from Rome, from Lazio and Tuscany, in a study performed with Sapienza di Roma, we found that some human donors, they were positive molecularly for Leishmania tarentule. And then in a study, parallel study in Linosa Island, here in the southern part of Sicily, while we were running an eradication program for Dilophilaria, we also took samples of dogs and humans for Leishmania, and we found them positive for Leishmania tarentule. So we had some evidence that maybe humans could be infected with this non-pathogenic species. So of course, considering that the main reservoir of Leishmania infantum is the dog, we wanted to see if also dogs could be infected or exposed to this species. And this started again in the province of Lecce, here in Apulia, where we wanted to see in a dog shelter where we had a lot of reptiles occurring in the surrounding areas, a lot of sandflies already recorded in previous years, and many dogs, if there was a possibility of sympatric occurrence within the animals of both species. And what we found is that dogs were exposed serologically, of course, which is by immunofluorescence. It's highly sensitive, but has a low specificity. So we had cross reactions for both species, but we did found some dogs molecularly positive in blood for Leishmania tarentule. And also, importantly, Leishmania tarentule infantum was detected molecularly in sandflies, such as Sargentomaya, which was the most abundant species, and they were feeding mainly from mammals and mainly from humans. So what we know thus far, or to this moment, is that the Leishmania tarentulae has a wider distribution. It may be possibly also already, as in the case of Leishmania infantum, already also occurring in Southern America, given the translocation of its typical host to that region. But also it's been already isolated from many reptiles and sandflies around the Mediterranean basin and even Asia. So this study we published 2 years ago in the Transboundary. We also have the cover of this issue, and so what the main result or main outcome we had of this type of studies is that we need to consider as a whole the epidemiological context, including different type of sandflies, not only phlebotomous sandflies, the implication of other permissive and non-permissive hosts, humans and dogs occurring in an epidemiological context. So in this case, one of the last studies we performed on this area was here in the University of Bari, in our campus, which is near this municipality of Valenzano. So we, in this site and also here in another near area, which is a dog shelter, a small dog kennel, we collected samples from reptiles, from dogs, and from sandflies. We did an entomological service, and we were able to isolate Leishmania tarentulae from geckos and sandflies, which will be used to understand furtherly why the species is not pathogenic and what are their biotechnological applications. For example, if we're considering to use it as an immune therapy, given that it did infect macrophages of mammals. In this case, what we found is not only that we had some typical trypanosomatids, such as Trypanosoma brachydactyly, but we did find also geckos infected with Leishmania infantum, and we found Leishmania amastigote-like forms of Leishmania infantum in the bone marrow of these geckos. And these isolates are now being studied to see if and to further verify the immune response Leishmania tarentulae has in vitro models. I mean, in PBMC, in macrophages and monocytes of dogs, to see if we have a polarization to the Th1 response, which is a protective response given by a cellular part of the immune response. In this case, just to wrap also the idea of what our studies mainly focus on regarding reptiles and their parasites. If we consider the main aspects that I mentioned at the beginning, hepatological medicine and the parasitology of reptiles, we can also consider the possibility of controlling and formulating new strategies of prevention for zoonotic parasites. I mentioned before that the snake mite, it's an important vector and has many deleterious effects in snakes, but I didn't mention that this mite, when there's absence or hyper infestation of these mites, they can also feed on humans. So it can be also implied or considered as a zoonotic parasite. And it can transmit, for example, Aeromonas mechanically to humans. In this case, the treatment, it's very, there is a few studies on how it can be controlled. But the snake mite, it's very important in mainly in large collections of snakes. It's called Ophionisus natricis. It has a worldwide distribution, and it's mainly found in snakes, in different type of snakes, but generally in captive settings, but also in some cases, for example, in Australia, it has been found already in wild species of reptiles. This mite is parasitic in its adult phases and as protony, but we can find in the environment the larvae and the deutonyms. And importantly, it can go up to 3 months without feeding, so it can survive in humid areas and up to 30 degrees of temperature for a long time. If it needs to do a hypobiosis, it can stay in the environment, and then when there's a suitable host, it can go again and feed on. What are the implications? It's found throughout the body. It feeds, and then the female goes to the ground and places deposit their eggs, and then it goes again. So it does many gonotropic phases, and it's seen mainly in the dorsal area and in the gular area of snakes. And it has many direct effects, such as dermatitis and dysecdysis, but even it can cause death when it has a hyperinfestation of these mites, because it also produces pruritus, and snakes, of course, do not have legs, so they cannot scratch. So many of them, they will be for a long time inside the water bowl, to try to kill the snake mites by drowning. So they will have some behavioral changes. They can become more aggressive because they are very irritated because of the constant itching and pruritus caused by these mites. But most importantly, they can transmit different pathogens, such as I mentioned before, the inclusion body disease virus, and they are also being implicated in the infestation of humans in different areas, of course, mainly those people that work directly with reptiles. How can we treat these mites? Generally, there's a multiple approach in which we try to take care of mainly of the infesting stages, but we have to also consider the stages that are in the environment. Consider that they can live up to 3 months without taking blood. So there are many ways, such as, for example, water baths of the snakes with the povidine, cleaning profusely the enclosure, using biological control with other mites that are predators of the snake mites. Or we can use, for example, different type of drugs, such as the chlorobus fipronil, other pyrethroids, and even macrocyclic lactones, which have some risks, all of them, for the use in snakes that are very sensitive to this type of compounds. But in many cases, or almost in all cases, it's almost impossible to eradicate just by or using this approach. So we wanted to try to use another type of approach, considering the available insecticides and acaricides on the market. So we considered to use imidazolines. As you may know, they are used mainly for the control of many ectoparasites in dogs and cats, such as ticks, fleas, they control those slow feeders mainly, but they also kill, for example, after the blood meal. So they do not avoid the transmission, for example, of leishmania, but they do kill afterwards the sandflies. And there has been some studies in snakes where they have seen that they have it's useful for the treatment of ectoparasites, mainly in ball pythons and Burmese pythons. And of course, one of the great advantages of the isoxazolines is that it has very few adverse events or effects in even in mammals, and it can be used in large doses without producing any type of clinical signs. So we use NexGard or Afoxolane. I'm not giving any publicitary advertisement, but this is a study where we use this type of drug that it has the claim for fleas, ticks, mites of different types, mainly in dogs. So we use it to see if it can be in an off-label study for snakes. So what we had is the opportunity to apply this strategy because of a hyperinfestation in a site here in Sicily called Girone, where there were two sites, one from a breeder and one from a owner of some snakes. in which they were infested both with they were friends, so one of them lent to another a snake for reproduction, and so the python that he had introduced to the other side the mites. And as you can see here, we had a high infestation in one place, 33%, and in the other almost 80%. And they were from many type of snakes, from 6 genera, almost 16 species. And they had different dermatological signs associated with the presence of mites, such as discectitis and dehydration. And even some, they had almost scale rot, which is a bacterial dermatitis in snakes. And many of them had mites in the typical areas where they can, of course, produce negative effects. And they had clinical signs associated to this type of infestation. So what we proposed to the owners was to eradicate by treating all of the animals, so not leaving any animal untreated, in order to avoid the 3 months of environmental mites that could afterwards feed on the untreated animals. And just that, so then we can verify that was only the effect of this afoxolaner. And then we treated them orally in most cases, and some of them we give for the smaller ones, or for example gravid females, we gave them on the prey, feeding or giving in the mouth of the dead prey already the afoxolaner. We took blood to have the plasma of these animals to also assess if they were having the plasma concentration at the proper moment and how long it was maintaining plasma, to see we had the coverage that we also see, for example, in dogs. It's for 1 month, more or less. So here you can see how we gave the afoxolaner. Of course, it was dosified depending on the weight of the animal. And the good thing of the presentation of the tablet, of the chewable tablet of afoxolaner, is that it's highly absorbable. So within a couple of hours, it's already digested in the stomach of the snake. And here you can see, for example, in a small Lampropeltis or king snake, we place the afoxolaner inside the mouth of the dead mouse, and then it was fed, and it also worked as it was absorbed quickly after the ingestion of the mites. Afterwards, then we did mass spectrometry, and what you can see here is that we had at 24 hours the peak of concentration, and it remained at the efficacy concentration till day 28. So it covered more or less the 30 days or the month of application of the afoxolaner as in dogs, with no side effects. The good thing is that after almost T1 or after 14 hours, all the snakes were with no mites, and they remained with no mites until day 28, and they went from having clinical manifestations or dermatological signs to being apparently healthy at day 28. So of course the recommendation would be to treat them for 3 months if we have an infestation, to avoid those mites that are still in the environment to be able to feed on untreated snakes and then of course die, and to do a treatment if we are not able to see mites once we acquire a new animal to be integrated to a collection during quarantine, to avoid then to bring other parasites in the sense. So deworming, and also a chewable or a systemic insecticide. And this way we can avoid these parasites. Now, to finish with some of the examples of parasites that can be of importance, I will show you 2 that are related to trophic transmission that are foodborne parasites. And as you can see, we have a large group of parasites that can be transmitted orally, but those of importance are more important are within the cestodes order and pentastomids. And if we consider Europe, there's still many gaps, but for example, sparganosis, which is produced by Spirometra erinaceae europaea here in Europe, is transmitted by the ingestion of raw meat of mainly snakes, but also, for example, toads, and is transmitted mainly to cats. So cats are the definite host of this cestode, and of course, they are the ones that perpetrate this cestode within urban area, but it can also infect humans if they are in contact with the intermediate hosts. And it's a very pathogenic disease because the perocercoid or the metacestode of these cestodes, they can migrate to different areas. For example, if they go to the eye, it's ocular sparganosis. If they go to the brain or the central nervous system, it can produce neural sparganosis. And the most pathogenic one is when we have Sparganum proliferum or proliferative sparganosis, which is the one that the spargana, they can replicate, as for example, for Tatiridea of mesocestoides. They can replicate and profusely and then invade and migrate to different areas. So it's very difficult to treat and to control. On the other hand, we have pentastomids, and well, it's a very rare-ish parasite here in Europe, but it's very common in other areas. The first time I saw this parasite was in Brazil when we were in a field trip activity, and we found this snake which was emaciated. It's a Botrus jararucusu, a venomous snake, a type of viper, which was emaciated, dehydrated, and died after capture. When we did a necropsy, we observed this vermiform, where like parasites, which are actually crustaceans or type of crustacean parasites, and they are pentastomids, which invade the lungs of these snakes or other different hosts. So they are very common. They are very pathogenic even to their definitive host. And when the snake dies, it's another Botrus jararucusu, they start to go out of the glottis of this of their host, and it can invade even wild animals, producing then their death. So it's still very pathogenic host parasite even to its typical host. Within pentastomids, we have, I think, the one that maybe veterinarians know the best is Lenguatula serrata, which is a pentastomid from dogs, transmitted by ingestion of mammals such as sheep, ruminants, or other mammals that are infested with the larvae and nymphs. And generally what occurs is that in snakes, for example, the adults live in the lung, and then they release the eggs, which are then ingested or go through the wall of the lung to the intestine, and then they are excreted. And then, of course, their intermediate host, it's infected by ingesting larvae or eggs, which go to the main organs, which are liver, spleen, and the mesenterium. So humans, they have exactly the same type of transmission if they eat raw or uncooked meat of snakes, if they are in contact with the liquids or fluids of snakes infected with the larvae or nymphs, or if they ingest contaminated, for example, water or spaces with the larvae or nymphs or eggs. And they will go mainly to liver, spleen, and mesenterium. And the symptomatology depends on the number of ingested larvae or eggs. There are some cases, for example, that what it's seen is that it generally seems to be a case of a tumor of a neoplasia, because it produces a type of granulomatose and caseous nodule where it encysts and then dies. So it's we are dead and host. The larvae doesn't develop to nymph and to adult. But in some cases, depending on the level of ingestion, it can produce very deleterious effects, such as, for example, this dog infested with Porocephalus crotale in North America, or this person infested with armilifer in North Africa. And the transmission, as I mentioned, it can be also not only by eating meat, but also with the water or handling snakes that are infested. So we have to be very careful. Many of their typical species that can be infested, such as large boid or python snakes. And in generally occurs asymptomatically, but once the larvae and nymphs die, then we can have different clinical signs, which are generally associated to they are generally associated to the number of larvae, for example, this case, a lot, or when the human patient is going to go under surgery or has, for example, a routine radiography and they observe the encysted larvae or nymphs. Well, just to finish my story of today and this conversation, I would like to give you two examples of the area now that we are more focusing on, considering all the elements that I gave you today, which is ethnoherpetoparasitology. If we consider that we are in strict relationship with reptiles, we have other relationships that are cultural, religious, or depending on the area where we live on, that can be through that it's studied under the concept of ethnoherpetology, which is the use of reptiles for medicine, for magic, for different activities such as snake charming, or using this as a symbol, for example, in festivals. So we applied that to the concept of parasitology to have this wider concept of ethnoherpetoparasitology. And we started this concept with the study in Morocco, applying this concept in Morocco. In Marrakesh, there is a very large market and plaza called Jema El Fna. And in this plaza, you can find not only snakes, but also a lot of reptiles used for magic or for medicine, traditional medicine. So this is the plaza, the square. There's a mosque over here, and this is the souks of Marrakesh, also very famous and very big. And within these souks, you can find chameleons, parts of reptiles, turtles, tortoises, and other reptiles used as pets or as for traditional medicine. And if you go to the center of the square, you will find the snake charmers that have different type of snakes that go from mildly venomous, such as the Montpellier snake, to highly venomous, such as the puff adder or the Egyptian cobra. So we, considering this unique epidemiological context where these animals were kept, brought from the natural setting to a urban setting, we wanted to identify parasites and pathogens that were present in these animals in their blood or their feces that were associated with the handling of these animals in this specific context. So we took some swabs from the oral cavity and from the cloaca of these animals, and also we did blood draw. Generally, as I mentioned before, from many snakes, it's possible to do the blood draw from the coccygeal vein, given that it's a wider vein, specifically if you're working with elapid or vipers or with colubrid snakes. It may be easy to access, and you can obtain a proper amount of blood that then was used or that was frozen or put in FTA cards that allow us to bring them and then process them molecularly. And then we look for most of the pathogens I mentioned today that are bacteria, some parasites, of course, Leishmania, and we also perform blood smears to see if they have any type of hemoparasites. And for the swabs, we did mainly parasites, protozoa, and cesto that we were very interested in looking for the possibility of finding, for example, spirometra or other zoonotic cestodes. And we found many interesting elements, such as, for example, gametes of hemoparasites that are specific for reptiles, but it means that they were infected in a natural setting, possibly by ticks or by mosquitoes. And we did found some pathogenic vector-borne bacteria, such as Anaplasma phagocytophilum, which is the human granulocytic anaplasmosis, which produces different clinical signs. So this is the first time it was found in snakes from Morocco, and it was associated to mainly mildly venomous snakes, such as, for example, this Montpellier snake. But we also find rickettsia, and we found a different type of rickettsia from the spotted fever group. And this is important because this means that these animals are indeed infested by ticks. Most likely they lose them after being captured, so ticks may be then released in the environment. And we found two types of Leishmania, two different animals infected with Leishmania tarentole. One, the first time of a snake in North Africa, which is the Montpellier snake again, and also a chameleon. And we found some cestodes mainly from snakes and also from Cryptosporidium, from this worm lizard, that it's a type of, it's not a lizard, it's not a snake, so it's in between. It's an Amphisbaena group reptile. And we did find many type of bacteria in the oral cavity and also in the cloacal cavity that can be associated to, if the animal bites, to secondary infections of bacteria, such as, for example, Pseudomonas aeruginosa, and importantly, Morganella morgani, which is a bacteria associated many times with snake bites, in which it's a dry snake bite, which means that the snake did not inoculate venom, but it did perforate the skin and thus infected the person with this bacteria, which can produce pathogenic effect in humans. So this is the first case or the first study. And then recently in Italy, we were able to participate from another event in where we, in this case, added another concept, which is conservation. We already discussed about parasitology, about the concept of One Health, studying the relationship between humans and reptiles, but we also wanted to add the element of conservation given by the relationship between humans and reptiles, also considering the bad rep snakes may have, as they are seen in many cultures as evil or having negative connotations. So in this small town in the center of Italy, in Abruzzo, called Cocullo, they use mainly this species of snake, which is a four-line snake, for a ritual is called Il Rituale del Serpare, that you may see here. So this ritual is a sacro-profane festivity that occurs every 1st of May in this small town, in which there are some snake hunters that are able to, or authorized by the government to capture different species of snakes, all innocuous snakes. And then they place them the 1st of May in the statue of San Domenico and parade them around this small town. And they are released again in nature. Given the cultural, traditional background, it's already been considered as a World Heritage element. Thus, the government and local authorities have been pushing for education and monitoring of these animals in order to protect them and to observe which is the impact this tradition has on the snake populations of this of the surroundings of this small town. So for more than 12 years, there's been taking place a project which monitors these animals. So each year before the festivities, the snake catchers need to come to the comune, okay, to the main town, main square, and the snakes are assessed by a veterinarian, which places a microchip on the animals to be able to identify if they are recaptured the next year. And they are measured, weighted, and seen if they have any elements. Sometimes they take some samples for bacteria and for some fungal diseases. And this has shown that some snakes have been captured for more than 10 years and they're still there. So in some way, these animals are protected because of this festivity, whereas in other areas, when a person sees a snake, they are generally killed on site. And last year we had the great opportunity to participate from this monitoring activity, and we included a more holistic approach. We try to acquire, also take some blood samples, ectoparasites, clinical examination, oral and cloacal swabs in order to see if these snakes had what was their typical fauna, because it was never performed in Italy for wild snakes and live wild snakes, and then which would be some elements that could be of zoonotic concern. And we were able to publish this year, in which we worked together with the local authorities in order to process more than 110 snakes last year. And we found ectoparasites. What you see here is a western whip snake with Mesostigmata mites, macronicid mites, and we found 2 species, Emirelepis piger, and also we found we're still working on this, a possible new species of Ophionisus mites from wild snakes. And of the sampling we were able to perform, we found for the first time, we did a coprological study on wild snakes on Italy, because there are some studies on pet snakes, but not on wild snakes. And we found different protozoarian species such as Imeria, capillary eggs, cestos, such as this is I think it's Imenolepis nana. And then we found some spurious parasites such as larvae mites that were, of course, ingested maybe through predation of some lizard or rodent. And regarding reptovector-borne pathogens, we found some rickettsia, which means that these snakes were also infested in one point, and most likely they are infested when they are during summer and spring, because this occurs a bit before the middle of spring, or not in late spring. And we found also again in the swabs and also in blood, in 4-line snakes and in western whip snakes, we found leishmania that most likely is Leishmania tarentule. So in this case, we further address that snakes also can be infested, and this is most likely because despite what is considered that snakes have a very thick skin, they can be a suitable blood source for sandflies. as you can see here. And sandflies can feed and they can thrive after feeding on snakes. So they can be also part of the epidemiological or the biological cycle of life cycle of different species of Leishmania. Of course, we still need to consider what type of species of Leishmania they may be reservoirs. So to conclude the conversation of today, consider that within the herpetological medicine and this concept of ethnoherpetoparasitology, the main figure we have also is the conservation of species. So then we are bringing more elements for the possibility of conserving important animals that have already an important role within the ecology of certain ecosystems. So the most important thing for conservation and even for the studies within herpetological medicine is the education. So we also, in many settings as today, we try to bring and update the information within handling, within medicine, and of course parasitology, and not only to bring the risk, but the prevention of zoonotic vector-borne and different zoonotic pathogens of parasites associated to reptiles in order to conserve the species. Because once we have knowledge, then we have the power to conserve even those less charismatic species, people that consider that are, for example, snakes. And with that, I would like to thank you for your attention. It's in Portuguese, I don't know why, but thank you, grazie mille, for your attention. In any case, if you want to follow our recent and our further studies, you can always follow us in Instagram in our account named Parasiterranean. Thank you so much for your attention. If you have any questions, I'm here. If not, you have my email and we can discuss further on.
Thank you. It was really interesting.
Thank you so
I have
much.
a question, if possible.
Yes, sure.
Are there any cases of, because we talked about the contact with between human and wildlife species of reptile, and also with captive animals, are there cases of reverse zoonosis in Italy or?
Of reverse zoonosis. Well, it's not been recorded as well as properly as it should be, but of course it can be mainly because given by bacteria, in the sense that if a person is has some type of bacteria, it can go to the reptile. This is mainly not by with wild reptiles. I mean, this could be the case, for example, if we're looking at a reptile that is kept as pets, in which in most cases is if the husbandry is not the proper one, they can suffer from low immunity and be susceptible to secondary infections. So if the person is handling and has one type of infection, or for example, what happens in many times, mainly with primates, if a person has a virus that for us could be non-pathogenic or not so pathogenic for reptiles, such as turtles, they are very susceptible for herpes virus. So in that case, it's not so studied, but it can happen, of course, in anthropozoonosis.
Thank you so much.
You're welcome. Any other questions? If not, I would like to thank you again, and hopefully we'll see each other in a near future when I come to Sardinia again. And if you have any ideas or would like to work in any case with reptiles, just write me an email and we can discuss further on.
Okay,
thank you so much.
Thank you.
Have a nice,
Thank you. Bye.
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 Have a nice day.
Bye.